Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Associate Director and Senior Principal Scientist Kyung-Ki Yang, Senior Scientist Laura Clemens, and Senior Scientist James Bowden will introduce you to the new features and enhancements available in DILIsym 11. So Dan, our business development director, is going to join us later for the Q&A, but I'll now pass things over to Kyung-Ki for the presentation.
Thanks, Jasmine. Yeah, so without further ado, why don't we dive in? And before we get into the DILIsym, let me pause here for a moment to introduce ourselves, like us at Simulations Plus. So we'll spend next hour to talk about QST model DILIsym. But at Simulations Plus, we actually offer a variety of different modeling and simulation platforms and also solution services, including like cheminformatics, PBPK, pharmacometrics, and all the way through adaptive learning, interactive learning tools, and medical communications. So just wanted to highlight, we are not just QST group, but we do more than that, like whole different solutions across the drug development and also post-marketing timeframe. And now on to QSP. So collectively within our QSP group, within Simulations Plus, we have more than 20 years of experience in QSP modeling, helping pharma companies and the industry like inform their decision making, important internal and also regulatory decision making by using our modeling approach to predict efficacy and toxicity of drugs or drug candidates. So we will talk about the DILIsym, but we have a variety of different platforms and the model, both QSP and QST models already developed and validated for prediction of toxicity and efficacy. For toxicity QST models, we have DILI, like liver injury, and the kidney injury models here. And we have the efficacy prediction models for metabolic disease, immuno-oncology, and other inflammatory, autoimmune, and fibrotic diseases. So just highlighting our capability at our QSP group. So with that, DILIsym, I would like to start with impact. So this is just very high level summary, but this slide speaks to the impact the DILIsym platform has been made for past maybe more than a decade. So we worked with almost 60 different biopharmaceutical companies to help with their internal and regulatory decision makings, mostly based on mechanistic modeling, as you will see more today. But the DILIsym has also been used to inform the hepatocyte loss based on the biomarker profile, and also inform the mechanisms underlying bilirubin elevations, which can be false positive signals. As far as I know, DILIsym has been used more than 50 like regulatory submissions and communications, and like 8 publicly disclosed drugs are there, like in the market, where DILIsym contributed to liver safety decisions. So the impact is there, and now what is it? Oh, before we get there, this is another recent paper published on hepatology by authors, like DILI experts, including like authors from FDA. And in that manuscript, the table 3 here has the summary of non-clinical data assessed by DILI team at FDA. And as you will see, DILIsym has been listed as the only QST platform for DILI assessment, again, reviewed by FDA. So we are very proud of that. And for those who are new to QST DILIsym, it seems that we have a mix of audience here. Briefly, so what is it? Within DILIsym and for QST approach here, we integrate drug exposure, toxicity mechanism, and physiology and inner patient variability related to underlying physiology to make predictions. The foundation of QST model, DILIsym here, is a mechanistic representation of underlying biochemistry, describing pathophysiology and mechanisms leading to hepatocyte injury. And as part of that physiology, within DILI, the inner patient variability is important because only a subset, most of the time, only a subset of treated patients develop some symptoms of DILI, right? So we need to account for those inner patient variability, which is represented within our virtual population, like SimPops. So that's underlying physiology. And to predict the DILI responses for a given drug, we need drug-specific exposure. In this case, hepatic exposure, the site of toxicity. So we use PBPK model to predict, most of the time predict the exposure, drug exposure within the liver as a driver of toxicity. And we have our good GastroPlus platform for PBPK modeling. And another key input is drug-specific information is toxicity mechanisms. So toxicity mechanisms are characterized by in vitro mechanistic toxicity assays to determine if and how drugs interfere with different toxicity DILI mechanism pathways and potentially leading to hepatotoxicity events. And here is the very high-level schematic of DILIsym. It's a mechanistic model, so it has multiple sub-models representing essential cellular processes. And of course, different sub-models interact with each other mathematically and also mechanistically. The key is hepatocyte life cycle model, right? We are simulating predicting liver injury. So we have the full hepatocyte life cycle model for 3 zones, including injury and hepatocyte death via apoptosis and necrosis, but also regeneration is part of it. So it's a full dynamic model which can predict injury and also post-injury recovery. And it represents key intrinsic DILI mechanisms such as oxidative stress, mitochondrial dysfunction, and bile acid transporter inhibition. And as you will hear more about it, we'll have updated our cholangiocyte and cholestatic injury model, as well as adaptive immune, like T cell submodel. And model also has representation of clinically relevant biomarkers, ALT, AST, total bilirubin, etc., so that you can put the model output in the context of clinical setting. We have multiple representation of different populations like humans, adults, and we've added pediatrics, which we will talk about today, as well as a few other preclinical animal models. So far, we have represented over maybe 90 compounds, and these are compounds with clinical data, which can be used to validate the model performance. And we had more than 80% success rate on predictivity with 0 false positive predictions. And lastly, the mechanistic model, as you can imagine, it's well suited to simulate and predict hepatotoxicity outcomes as a result of multiple mechanisms, the combination of different mechanisms, as well as combination of different drug therapies in the polypharmacy setting. And a little bit more about inputs and outputs of the model. key inputs are twofold: exposure, so any PK data that you would need to build and develop PBPK model would be needed, preclinical, in vitro, clinical data if available. And the other important input would be mechanistic data. These are in vitro mechanistic experimental data to determine the mechanism of toxicity as well as potency for those mechanisms. And inner patient variability is represented within the model. So within DILIsym, the simulations integrate exposure and the mechanistic interference. And model outputs are actually dynamic, like time course of hepatic injury and associated the hepatic biomarkers. And we also have the famous Yiddish plot as one of the key model outputs. So that's very high-level overview of DILIsym. Then on to the topic of DILIsym 11. So what's new? So we released version 11 back in May. So it's been out there a few months. And within this release, we have a few key scientific updates that might be of interest to you all. First, the new CD8 plus T-cell submodel for retrospective evaluation of CD8 T-cell mediated, CD8 plus T-cell mediated hepatotoxicity. And we've added new pediatric representations, which can be used to explore pediatric liver safety outcomes for certain compounds. And we've significantly updated and enhanced our cholestasis and bile acid representations. And in addition to different injury models, we also updated our adaptation models. For example, nerve 2 responses after oxidative stress. So that part has been added, as well as the new SimPops and exemplar drugs. So for the next 30 minutes or so, we'll walk through each of items here, and also there will be a demonstration of the software. At this point, I'll invite Lara to give an update or present our new T-cell model.
Great, thanks. So I'm very excited to introduce the CD8 positive T-cell mediated cytotoxicity submodel that has been introduced to DILIsym version 11. This T-cell model is built on the back of many years of work and leverages the existing framework within DILIsym. So if you look at this top figure, we can walk through the process from drug exposure to hepatocyte stress. Hepatocyte stress and cell death can lead to danger signals or DAMPs, and that can lead to what we would traditionally think of as innate immune responses or potentially to liver tolerance. And so this pathway has been in DILIsym for a long time, certainly including the innate immune responses to liver injury. And what's new is that we've included the adaptive immune response that may follow those initial triggers. And this adaptive immune response may then lead to additional cytotoxic T-cell mediated hepatocyte cell death. We do and build. And so how have we constructed this T-cell model? This has really been built in a staged approach, starting from all of the available mouse data, particularly for the experimental antigen ovalbumin or OVA. And this allowed us to characterize how CD8 T cells expand and contract and their resulting ALT elevation. So this allowed us to calibrate the underlying dynamics of our model and the level of cell death that we might expect from a cytotoxic mediated event. And then from here, we moved into then a drug representation. We used amidiaquin as our representative drug. And again, we started in a mouse model. And so this leveraged data that was available for PD-1 knockout anti-CTLA-4 mice. And these were very susceptible to then a amidiaquin-induced CD8 positive T-cell response that then led to ALT elevations. And so this allowed us to then bridge the connection from administering a drug to then having some antigen present in the liver that can lead to T cell expansion and then potential cytotoxic response. From here, we then translated this to a human representation of amidiaquin, leveraging available clinical case studies. And then we finally, if we do one more build, and so this is the model as it exists, and I'll talk a little bit more about it in the next slide. And we're very excited because this is now available in DILIsym v11. And I will highlight that this is a very exploratory model still. We've built this out for amidiaquin, and we're really interested to see how it responds with drugs that you all are interested in testing it with. But as of yet, it's a primarily retrospective model and is not yet considered predictive. So if we go to the next slide, I'll talk a little bit more about the submodel itself. So the submodel includes a full CD8 positive T cell life cycle. And so we have, for example, T cell activation, exhaustion, proliferation, apoptosis, and then importantly, we have antigen presentation by hepatocytes in the liver that's determined by drug exposure. And then we have apoptosis of the antigen presenting hepatocytes that's induced by contact with the CD8 positive T cells in the model. Critically, there's a lot of variability that one might expect in the way that the T cells may respond or in the level of antigen presentation by hepatocytes. And so we do represent this variability in both a human and a mouse T cell SimPops. And that includes variability in the life cycle, differentiation, cytotoxicity, dynamics of antigen uptake and clearance, and of course, avidity. And so you can see an example E-dish for our human T cell SimPops simulated with our amidiaquin representation. And you can see a range of possible responses resulting from individual variability in this pathway. And so you can use this submodel to explore different injury profiles and drivers related to T cell mediated drug-induced liver injury, and look at how different initial conditions or different initial kind of baseline characteristics may then drive different responses, and pair that with your novel compounds to explore what types of signals you may see, or retrospectively, what potential signals or what potential drivers may explain the signals that you are observing. And if you are interested in learning more, we do have a recent publication on this work, which can be found in EOMT.
Right. Thanks, Lara. Yeah, so it's very exciting that we've been working, as Lara mentioned, working on this T cell model, and it's very exciting that we have a version there. This is the first step toward addressing immune-mediated toxicity, hepatotoxicity, and idiosyncratic hepatotoxicity. So find out more about our T cell model within this publication. And then another exciting addition to DILIsym 11 is pediatric representation. So there is a great unmet need in terms of the DILI assessment, prediction, evaluation in pediatric population. And to tackle this unmet need by modeling and simulation, we have laid out a staged approach. So within DILIsym, DILIsym has been developed, validated, and used over and over for adult population. So building up on that, we've added the pediatrics population first, starting with creating small adults, meaning like the reproducing the height, body weight, organ volume, etc. Basically, physiologic size of the pediatrics. And then in terms of drug exposure in pediatrics, it's been already well addressed in PBPK specific platforms such as GastroPlus. So we do not need to reinvent the wheel here. So we will be leveraging the PBPK, the established PBPK, the software to predict pediatric specific exposure, and those exposure profiles can be produced within PBPK platforms and imported into the DILIsym. And within the DILIsym, the focus will be representing pediatric specific physiology and biochemistry related to hepatotoxicity mechanisms. So within our model, we have multiple different toxicity pathways. So we started with the bile acid model, basically look for the data for pediatric specific bile acid profile, the ontogeny of enzyme and transporters related to bile acid disposition. So that we have pediatric specific representation of the bile acid homeostasis. And then the next step will be the step-by-step to, again, pediatric specific representation of oxidative stress pathway, again, based on the data available, and then onto the mitochondrial, the bioenergetics, which is specific to pediatric population. And once we build up on all of that, we need to validate the actual DILI responses, right, by simulating drugs with known clinical DILI responses in pediatrics. So we have different blocks of puzzle or pieces of puzzles, and let's make sure when we put those pieces of puzzle together, we still capture the clinical responses. So that's our roadmap. So far, we've completed 2 stages of those roadmap. So within the DILIsym 11, we have added 4 age groups of pediatrics, representing toddler, preschooler, school age, and adolescent. And we have baseline and also SimPops representation with population variability. And age-specific representations of bile acid homeostasis was developed, leveraging available clinical data. Exploratory predictions in pediatric liver safety profile for bile acid-related mechanisms can be made using this version. And we are continuing to build upon the rest of the pediatric components, which we are hoping to release as part of our next DILIsym 12. And quickly, this is a poster presentation at past AAPS conference, which covers our pediatric representation so far, focusing on bile acid homeostasis, leveraging the pediatric specific enzyme ontogeny data, and making sure the simulated bile acid profiles in our pediatrics match those clinically reported. And one of the challenge here is the data gap in terms of clinical data, really comparing pediatric versus adult DILI responses for a given drug. So for bile acid, we've simulated an exemplar dummy drug, if you will, to qualitatively and quantitatively compare simulation results between adults and pediatrics if the bile acid pathway is impaired. Our initial simulations show that there are no significant differences in terms of bile acid in response to bile acid like B7 inhibition. But again, we will continue to look for clinical data to further validate this. And now at this point, I'll invite Jim to give an update on our cholestatic injury model.
All right, thank you, Kyung-hee. So another update that we're excited about is the cholestatic DILI representation in DILIsym 11. So this is basically an expansion of the already existing MDR3 inhibition model in DSX, or the preceding version, but we have added new science in DILIsym 11 related to this MDR3 inhibition. So basically, older versions of DILIsym has mainly been focused on hepatocellular mode of liver injury, whereas we recognize from the literature that about half of all liver injury cases can be attributed to cholestatic liver injury. So we're really trying to make a push towards, in a more detailed way, representing cholestatic liver injury in addition to the hepatocellular mode of inhibition as well. So one of the main recognized drivers of cholestatic liver injury is the inhibition of MDR3. So MDR3 is this transporter on the canalicular membrane of hepatocytes along with BSEP. Inhibiting MDR3 will basically lead to a reduction of phospholipids in the bile duct, which will reduce the ability of bile acids in the bile duct to form these mixed micelles, which will basically protect the bile duct or the cholangiocytes from bile acid induced injury within the bile duct. So that's how inhibition of MDR3 can lead to cholangiocyte injury. So we've been working on further expanding the representation of that within DILIsym 11. We've included some cholestatic injury-related adaptation pathways, including the cholhepatic shunt pathway for bile acids, biliary bicarbonate secretion. We've also now, relative to the previous version of DILIsym, now included new modes of MDR3 inhibition, whereas previously we've only represented competitive. Now we also have the ability to model mixed and non-competitive modes of MDR3 inhibition, which has turned out to be quite important in terms of its predictive ability. We've also have a non-MDR3 mediated phospholipid efflux representation. Not all of the phospholipids that are available in the bile duct are derived from this MDR3 mediated process. So that's also included within this updated representation, as well as including a more detailed cholangiocyte life cycle, including its regeneration upon injury. So these are all features now available in DILIsym 11, which has also been described in more detail in the article you see on your left-hand side, which was published in 2023. Part of this work also included a recalibration of our overall bile acid model. Since we are now kind of exploring in more detail how bile acids move into the bile duct, the overall systemic disposition of bile acids had to be reevaluated as well, which means the baseline structural representation for bile acid disposition had to be reevaluated, but also in terms of the previously available SimPops. So we've had to recalibrate the previous normal healthy volunteer SimPops, tested it with more than 30 clean and DILI-associated exemplar compounds that have some bile acid transporter inhibition role. Then those learnings were also carried over into our other SimPops, our NAFL and type 2 diabetes SimPops, as well as our rat SimPops. This also led to kind of an update that was necessary for our Sim cohorts, our smaller subsets of the larger SimPops. And this was also carried over into our new SimPops, our postmenopausal women or PMW SimPops. Next slide. And building off of that work, we basically also looked into more detail in specific MDR3 inhibitors. So we were able to identify two clean MDR3 inhibitors, those that have basically no clinically relevant cholestatic liver injury signals. These are loratadine and chlorpheniramine. And then two DILI-associated compounds that also are MDR3 inhibitors, itraconazole and verapamil, which are associated with cholestatic liver injury biomarker elevations, such as alkaline phosphatase and gamma-glutamyl transferase elevations. So we focus on these four compounds, two positive, two negative controls, to further validate our MDR3 inhibition model. So the simulations that we performed show that hepatic exposure, the MDR3 inhibition potential, and the mode of inhibition, again, competitive versus mixed versus non-competitive, really matter in terms of predicting cholestatic liver injury for these various compounds. And then we also came with a new kind of default setup for novel MDR3 inhibitors, where a mixed inhibition with the so-called alpha of 30, so not purely competitive, not purely non-competitive, but somewhere in between type of inhibition is currently the recommended mode of inhibition for a novel MDR3 inhibitor. So we're continuing to explore this, adding more compounds to our database to further validate this representation. But for now, in DILIsym 11, we have confidence in these 4 compounds, these 2 positive and negative controls, and to make this recommendation for more novel MDR3 inhibitors in the pipeline. Next slide, please.
Thanks, Jim. Yeah, so in addition to those additional injury mechanisms or updated injury mechanisms, we have expanded or updated our adaptation mechanisms as well. So injury is, again, with the DILIsym via multiple different mechanisms predicts injury, hepatocellular injury, and plus cholestatic injury too. But we know physiologically, in response to those initial injury, the liver triggers adaptive responses to protect itself, right? So that's part of like physiological dynamics. So within DILIsym, we have actually been implementing those adaptive mechanisms as part of the dynamic hepatotoxicity simulations. For bile acid, accumulation of hepatic bile acid activates nuclear receptor FXR, then which next subsequently decreases bile acid synthesis, upregulate hepatic efflux transporters, and downregulate hepatic uptake transporters to protect liver from buildup of toxic bile acids. So those FXR-mediated adaptation is part of the default bile acid homeostasis model. So it's routine part of the DILIsym model simulation. For mitochondrial toxicity mechanisms, again, there are numerous reports out there. The evidence of the increased mitochondrial biogenesis in response to initial mitochondrial injury, again, the protective adaptive mechanism of the liver. So we have added representation of mitochondrial biogenesis in past versions. But in terms of, so the mechanisms, equations are there, but in terms of the parameterization of those biogenesis response, those parameters were calibrated to the clinical data for a single compound. So with limited clinical data used to inform parameterization, it's an optional setup that users can turn on if needed for exploration. So this is in the future, we are hoping to identify more clinical data to further validate this biogenesis parameter. And what's updated in the current version 11 is NERV2 response in response to oxidative stress. So NERV2 activation induces synthesis of glutathione and antioxidant enzymes. Again, another way to protect liver from those damages. So we had in the past version DSX equations and representing the pathways of NERV2, but those are not fully calibrated in terms of parameterization. So recently we've leveraged the data, clinical data from acetaminophen to further enhance and parameterize those NERV2 related adaptive parameters. So those NERV2 parameters, updated parameters are now part of DILIsym 11. But as in the case for the biogenesis, it's been again tested with a single drug acetaminophen. So we made it as an optional simulation. The user will be able to opt in NERV2 adaptation as part of simulation to explore the impact of these adaptive pathways. And a little bit more on this NERV2 that's updated in DILIsym 11. So in the past, there is oxidative stress within the oxidative stress mechanism, right? There is always generation and clearance of oxidative stress, which balances out. And we used to have the linear ROS clearance pathway. Now that in the past version that has been updated to more mechanistically represent like non-enzymatic, the small portion of the clearance, and enzymatic clearance of the oxidative stress. And this enzymatic clearance is a pathway which is upregulated by nerve to signal in response to oxidative stress. Specifically, increase generation of antioxidant enzymes, as well as enhance recycle of oxidized enzymes to the reduced form. So we had updated this mechanistic pathway, and in DILIsym 11, the parameterization has been updated, leveraging, again, acetaminophen data. So this is a data set where acetaminophen was dosed repeatedly at therapeutic and supratherapeutic doses. And in the absence of nerve to response, the toxicity responses at supratherapeutic doses, 6 milligram per day, 8 milligram per day, are over-predicted compared to the clinical data. So the nerve to parameters were optimized to recapitulate the hepatotoxicity responses to this repeat supratherapeutic dosing of acetaminophen. So details of this recent APEP modeling have been published on CPT PSP, so you can check out those manuscripts. But yeah, this is basically the data set that were used to update the nerve to parameterization within DILIsym 11. But again, still only one drug, single drug was used. The clinical data for a single drug was used for the parameterization. So the users can opt in this nerve to effects into simulation if necessary for exploration. Okay, and next is, we have added quite a few new exemplars within the DILIsym. Amodiaquine as an exemplar for T-cell model and 4 MDR3 inhibitors, as well as newly updated acetaminophen models. And in both steps, our application case, which has been published in CPT in the last year. In addition, we've expanded our SimPops representation to pediatric SimPops, and as Jim mentioned, we've added post-menopausal women SimPops, as well as human and mouse T-cell SimPops, and the adaptation SimPops including those additional parameters for adaptation, and then newly updated acetaminophen immediate and extended release formulation SimPops. So I'm pretty sure you're all curious to see what it looks like in the DILIsym 11. So at this point, I'll invite Jim to give a demonstration of the DILIsym 11.
Thank you. Let me share my screen here. Let's see. All right, Jenny, can I get a thumbs up that you can see my screen?
Yes.
All right, perfect, thank you. So yeah, let's do a relatively quick demo of what DILIsym 11 looks like and some of the new features, where are they located, and let's run a few example simulations and look at some of the results so you can kind of see what these new features look like. All right, first we're starting the application. So first we have then the GUI that's opening. We can maximize the window here. So we have our application settings button at the top. Next to it, we have our DILIsym V11 documentation hyperlink as well. And it brings you to share file. So that's a new location where we store our DILIsym documentation. Then we can kind of walk through the various icons here on your taskbar on the left. So let's first take a look at the SimSingle window. This is where you can either create or import SimSingles, basically setting up your entire parameter sets for your species, your drugs, your time parameters, so on and so forth. Then our SimPops feature, where you can basically start or set up your SimPops simulations. Our run list, where you can create batches of different SimSingles or SimPops runs that can be run one after another. Our sweeps, where you can run through like basically a parameter sensitivity analysis. We have our monitor feature where clinically relevant and monitoring protocols can be implemented, such as stopping dosing when ALT rises above a certain threshold. Then you can create your own sim cohorts from existing SimPops in the sim cohorts feature. You can create your own output panels to kind of reduce the final size and output selection for your various simulation runs. You can create your specified data templates under the spec data feature. This is, for example, where you can import pharmacokinetic profiles that either have been derived from actual data or predicted from PBPK software such as GastroPlus. This can be then input into some of these DILIsym simulation runs. We have our toolbox with various tools for the user to explore. I won't go into detail here today. So let's take a look at sim singles and actually create one, like a dummy example sim single. You can create both DILIsym or MitoSym sim singles. For this demo, we'll focus on DILIsym. We'll give this a name, DILIsym 11 demo. So let's kind of walk through the various options here. So you can, we have our species parameters, which is by default set to human. You can see some of the other options here. So we have various human, mouse, rat, dog species sets. Ones highlighted here in yellow are the ones that have an updated cholestasis and bile acid representation, as highlighted before. Then we have our new pediatric representations, the 1, 4, 10, and 14-year-olds. And then we have these new CD8 plus T cell representations for both human and mouse, and an updated NERV2 adaptation response representation as well. If we go to the customization option, we can also look at some of these species-related parameters grouped by subgroups. So we can take a look at the subgroup option. So where can you find some of these new parameters for these new scientific updates? Well, under the biological specification subgroup, you can find some of the new pediatric parameters. If we scroll down, we can find our new cholestatic liver injury related parameters under the liver bile acid transport, phospholipid regulation, and cholangiocyte life cycle subgroups under the species parameter set. And then for the CD8 plus rate reg and basal levels subgroups, you can find some of the new CD8 plus T cell parameters. And then finally, under the nerve response adaptation, this is where you can find the new NERV2 parameters. And under the HC or hepatocyte antigen regulation subgroup, you can find some additional CD8 plus T cell parameters. All right, so let's move down the list. So we have our calories options here. I'll open that later. Let's focus first on the drug parameters. So here we have some new options as well. For the blank T cell submodel on option, which is basically a blank drug template in support of the new CD8 plus T cell submodel that you can select for exploratory purposes. The new APAP formulation exemplars that Kyung-hee mentioned are shown here as well for the CYP-IR, immediate release, and extended release ER drug exemplar sets. And then what Laura alluded to, we have new exemplars for amodiaquin. So we have 3 sets here for human with different levels of antigen presentation. And this is all in support of the new CD8 plus T cell submodel. We scroll down, we have some of the new MDR3 inhibitor exemplars, such as chlorpheniramine, emvodostat, which Kyung-hee mentioned. There's a recent publication on that specific exemplar. And then some more MDR3 inhibitor exemplars, itraconazole, loratadine, another MDR3 inhibitor, one of the negative controls, verapamil, one of the positive controls for the updated cholestasis model representation. And then we also have the amodiaquin exemplars for the mouse representation. And finally, we also have ovalbumin antigen representation, drug file, basically in support of the new CD8 plus T cell submodel as well. And that concludes the new additions to the drug parameter sets. Here again, we can go to customization to explore some of the subgroups related to the drug parameter sets. So we have the different modes of MDR3 inhibition now available under the phospholipid canalicular efflux subgroup under the drug parameter sets. And if we scroll down, we have the mechanistic intervention subgroup that previously existed, and this is where we've added the new T-cell model switch as well. And additional T-cell model parameters are also under the hepatocyte antigen reg subgroup under the drug parameter sets. And then we go to the gear icon, you can basically go to the mechanism selection feature. We have different so-called scaffolds represented in DILIsym, compound W, compound X, compound Y, which can be selected here. Multiple scaffolds allow you to potentially run toxicological DDI simulations. And under the select mechanism option, we have basically the same types of parameters previously mentioned, but organized under this mechanism selection feature. So some of these T-cell model parameters can be found under the antigen presenters mechanism. And some of the additional modes of MDR3 inhibition are also available here under the inhibition of MDR3 mechanism. All right, so then moving down the list, so we got our solver options. I won't go into detail there. You can select your output panel that either choose one that was previously created or customized by yourself. Then you can select your clinical monitoring selection if you wish to do so. Then we have our compound W, compound X, and compound Y dosing parameter sets, which are mainly used for our DILIsym or built-in PBPK representation. So separate from the GastroPlus specified data import approach, there's also a way to kind of representing your compound using the built-in PBPK representation in DILIsym, which is particularly the case for some of the legacy compounds in DILIsym that a user can, which a user can select as well. Then for the time parameters, you can select the total simulation time for your run, whether or not you're interested in running your simulation for just 1 day, 1 month, 1 year. This is where you would define that. Then under specified data, this is where you would select your potential previously generated PK profile from another tool, such as GastroPlus, and use that as your exposure for your hepatotoxicity predictions. However, you can also specify other types of state variables that are not exposure related, but that's usually for more advanced applications, but it's certainly possible. Then there's also a way to choose different initial conditions to go with your SimSingle setup. So let's create an actual example SimSingle for basically to represent the T-cell model exploration. So let's create a DILIsym SimSingle. We'll call this, for example, the mouse amodiaquin demo example, to kind of see what we need to select to run this amodiaquin exemplar under a mouse species parameter condition. All right, so that SimSingle is created, as you can see the left top of your screen. So now we'll kind of have to customize our selections. So we'll choose, instead of human, we'll go with one of the predefined mouse species representations from the MAC 2015 study that was actually used for the development of the T-cell model within DILIsym 11. So we'll use this for this particular example. We do not need meals for this particular example, since this is mainly used for mitochondria and bile acid related simulations. So we can turn off the meals. For drug parameters, we'll select will actually filter to kind of or accelerate the search for our particular exemplar. So the mouse amodiaquin, and then the ones that are offered will show up. In this particular case, we'll use the 30% antigen presentation option. Since some of the T cell related outputs are not part of the common outputs panel, we'll select for this particular purpose an all variables output panel, so that we can capture all the T cell as well as all other variables that are represented within DILIsym. So we can plot some of those T cell related ones later on in the plotting feature. For compound X dosing, we'll select our amodiaquin 10-week dosing protocol. So amodiaquin is basically linked to the scaffold compound X. We'll increase the simulation time from 1 day to 60 days. And we're basically good to start this baseline mouse simulation. So let's go to run SimSingle. It's saving, it's basically setting up, getting everything ready to run the baseline simulation. We'll hit start. And I'll just accelerate this a little bit for demonstration purposes. So the simulation is done after like half a minute, but since we stored all the variables, it takes a few minutes to store all the various variables. So within 5 minutes, all those more than 1,000 data or variables are all stored, so we can look at the results. So you can see we have 2,668 variables to choose from. So in this particular case, we'll look at time on the x-axis, and we'll look at some of the outputs of interest here. So plasma ALT. So how did this mouse-specific simulation, what type of ALT elevations did it produce under these T cell type conditions? So let's look at ALT on the y-axis. So you can see here after like a week or two, we really start to see an increase in ALT with this specific setup we created for this demo. So now you may be wondering, like, what led to this hepatocellular injury? So we can look at some of the T cell specific other outputs to see what may have led to this ALT elevation. So we'll clear this ALT plot. We'll go back to our text box. We'll type in total effector to get our total effector CD8 plus T cell option. We'll select that one. And we'll plot this one. So you can see this produced a very similar curve with a slightly earlier occurrence than what we just saw for plasma ALT. So this is kind of the preceding event that led to the hepatocellular injury. So we see this time-dependent increase in total effector CD8 plus T cells. So then you may wonder, well, what about the hepatocytes themselves? So we'll look at total liver fraction of antigen-presenting hepatocytes. And you can see even earlier, before these injury events happen, we see the fraction of the hepatocytes that actually present antigen started to climb very early on in the simulation. So this kind of gives you like a time-lapse view of the sequence of events that's happening that ultimately led to the biomarker elevation, all starting from this increase in antigen-presenting hepatocytes. So we'll click out of this and look at one other quick example. So we'll create another DILIsym SimSingle. In this case, we'll look at an example for the cholestatic liver injury model update. So we'll look at a specific MDR3 inhibitor, human itraconazole demo. So this is actually a compound that inhibits MDR3, but also BSEP. And we'll keep our human parameter set. We'll keep our calories set to human because now we are engaging the bile acid model, in which case we always recommend selecting the human calories for human simulations. So for the drug parameter set, we'll select our itraconazole exemplar that has been now added to DILIsym 11. So we'll select itraconazole. We'll update our time parameters from 1 day to, let's say, 3 weeks. In this case, we have actually developed the PK representation in GastroPlus. So we have a previously created specified data template that we'll select and kind of link to this itraconazole sim single. In this particular case, we'll use the baseline human, so a single kind of average representation of the PK profile that will link to this sim single. Although I'm using a single profile, I am going to now link the sim single to a sim cohort. So I'm going to basically start a SimPops configuration where the itraconazole sim cohort demo name is given. I'll provide the, so the base sim single that will be used here is the human itraconazole demo. Then the SimPops that will be selected will actually be a sim cohort just for demonstration purposes. But before I select it, this is also a good opportunity to show you where some of these new and updated SimPops are residing. So the new pediatric SimPops are located here, one for 14, 10-year-old SimPops. We have our APAP SimPops with additional PK variability for our APAP disposition. We scroll down, we have some additional updated SimPops and sim cohorts. Our new SimPops, PMW, as well as the NERV2 biogenesis SimPops, and then updated rat SimPops, as well as a new mouse and human T-cell SimPops. So for this example, for itraconazole, we'll select a sim cohort, basically the multi-16A, which is a subset of approximately 16 sensitive, there's some non-sensitive individuals as well, but mainly sensitive individuals from the larger SimPops. So now we have our sim single and our sim cohort paired together for this SimPops run. We'll just use the common outputs panel, and now we're ready to run this SimPops configuration. I will hit start, and again, I will accelerate the simulation here. So after a few minutes, we start seeing individuals completing their simulation. They either did not survive or did. So the color codes indicate whether or not an individual has not yet started a simulation, whether it led to a early simulation termination or basically simulated death, or whether the individual survived until the end of the simulation. So we can look at results. The Irish plot can scroll out of this initial view because there are actually 3 individuals that ended up in the Hy's Law quadrant at the right top of your screen. So we can take a look at some of the other outputs. Let's take a look at plasma ALT. Let's confirm these 3 individuals that showed up in that Hy's Law quadrant. Indeed, we see these 3 individuals with very high levels of ALT. What the plotting feature allows you to do is also click on individual profiles to identify which individual in the SimPops or sim cohort is the profile of interest. So we have patient 9 here. We got our patient 10, and we also have our patient 5. So that's one thing you can do here. Let's reset and look at another exploratory output. So instead of plasma ALT, let's look at some of the more cholestatic liver injury relevant outputs. So let's look at alkaline phosphatase. So with this MDR3 inhibitor itraconazole, we see elevations in plasma ALP. And
I think for the interest of time,
okay.
Let me take it back from here. But
Okay.
really nice demonstration of the software feature, especially the newly added DILIsym mechanisms. So thanks for the demo. And I will quickly share screen. We are at the top of the hour, but again, we've also all seen the demo. But just want to highlight, if you are interested in trial or have any questions, come to our webpage and make a request. If you are not a modeler but still interested in collaborating using the model, we can do a project in collaboration with you. So again, come to our webpage and make a request there. Finally, we will be having an actual tutorial in a month from now. So if you're interested, please register for it. At this point, I will turn it over to Jasmine and Dan to wrap up.
Well, thank you, Jasmine, and sorry for my tardiness. We have a couple of questions. If you have any other questions that we can't get to in the next minute and a half, please forward them and we'll follow up after the call. But I guess maybe the one question that I would ask is, it seems like you guys were very excited about the new SimPops being added to DILIsym. What if the user wants to add their own or create their own SimPops? Can they be accommodated, or do we have to wait for another version?
That's a great question. Definitely users can make their own SimPops and import it into the DILIsym, given all the data and physiological data available for that specific population. So short answer, yes.
No, very flexible is what you're telling us.
Yes.
Okay, great. And then maybe the only other question I had was, what types of data are needed to really explore the hypothesis regarding T-cell mediated hepatotoxicity, if I could say the word, using DILIsym? What kind of data do you need?
Sure. So some of the classic data that you'd use in your typical DILIsym examination, looking at drug exposure and understanding the underlying mechanistic toxicity, if any, is really relevant. But then since this is really well suited to a retrospective analysis, we'd also really encourage you to have the liver safety profiles of patients that maybe demonstrated liver injury signals that might be consistent with idiosyncratic DILI, and that way you can use the software to evaluate what types of patients that you're simulating may look like the patients that you're seeing in the clinic, and can you interrogate this for underlying drivers.
Okay, that's great. Well, again, given the fact that we're well over, I'm going to throw it back to Jasmine for a conclusion and thank our speakers for a marvelously informative talk.
Thank you all. I just wanted to remind everyone that this webinar has been recorded for playback and will be available on our website and YouTube channel. We've also put a couple links in the chat to visit us on our website, a link to the upcoming training, and then if you want to follow us on social media as well. This concludes our webinar for today. Thank you, everyone.
Thanks. Have a good one. Bye.